Explainer. Welcome, focus learners. Today we're tackling the absolute foundational framework of all life sciences. I'm talking about the biological method. I'm going to be your guide for the essential step-by-step facts and definitions you really need to master this topic, especially if you're gearing up for standard textbook board exams. We're laying down the baseline facts today.
And I am here to add a little chaos to that order. Because, while the textbook makes science sound like this perfectly straight, neat line, I'm going to show you how these concepts actually survive in a real, incredibly messy laboratory. We'll be digging into independent variables, control groups, and how data actually behaves when you put it to the ultimate test.
So, here's our roadmap for today. We'll be covering 6 essential concepts. 1. Defining the biological method. 2. Observing and hypothesizing. 3. Designing the experiment. 4. Gathering and analyzing data. 5. The role of skepticism. And finally, 6. Scientific theories and laws. Let's hit the ground running.
All right, Section 1, the biological method defined, the step-by-step approach.
Okay, before we get to the lab, we need to know exactly what it is we're talking about. So here's the exact textbook definition you really need to memorize. The biological method is simply the scientific method applied specifically to biological problems, solved through a structured, step-by-step approach. It's literally the engine that has driven scientific research for almost 500 years, going all the way from Galileo's experiments in the 1590s straight through to modern medicine and ecology.
Okay, sure, but that textbook definition is a little dry, right? In reality, the biological method only kicks off when a scientist encounters a real, tangible anomaly in nature. A biological problem isn't just some boring exam question, it's a mystery. It's like a biologist walking into a field and noticing, hey, why are the plants in this one specific patch suddenly growing twice as tall as all the others? That anomaly triggers a question, and that question is the spark for the whole shebang.
Section 2. Observations and hypotheses, forming initial ideas.
Once we have that spark, that biological problem, we move into formulating our thoughts. Observations use our 5 senses: vision, hearing, smell, taste, and touch. But they fall into 2 very distinct categories. Qualitative observations are descriptions, like noting that a solution feels cold or looks blue. Quantitative observations, on the other hand, involve precise measurements, like recording that the freezing point of water is exactly 0 degrees Celsius.
And let me just interject right here, because if you step into a real lab, quantitative data is the absolute gold standard. Why? Well, because saying it feels cold is totally subjective. My cold isn't your cold. But 0 degrees Celsius, that is invariable. It's measurable, it's consistent, and most importantly, it's strictly recordable in hard numbers.
Exactly. So the crucial point is that once you have your observations, you form a hypothesis. For your exams, definitely remember this definition. A hypothesis is a tentative explanation of observations that can be tested. It should be a general statement, it should be kept simple, and it absolutely must agree with available observations. Essentially, think of it as an educated guess that just hasn't been proven yet.
Right. But an educated guess just isn't enough on its own. You have to translate that guess into something you can actually test in the real world. This historical quote brilliantly illustrates how we do that through deductions. Lab scientists turn a vague hypothesis into a strict if-then logical statement. Take the historical search for the cause of malaria. The deduction was, if Plasmodium is the cause of malaria, then all persons ill with malaria should have Plasmodium in their blood. Boom. That gives you a definitive, testable target.
Section 3. Designing the experiment. Testing the hypothesis. Let's move to the real crucible of the scientific process. For your tests, it's really important to differentiate between an observational study and a true experiment. In an observational study, you're passively recording data without any intervention at all. But an experiment, that is an active manipulation of variables.
Yes, experiments are where we really get to work. This is where we impose a treatment. We actively change an independent variable, control literally everything else, and measure the dependent variable. It's honestly the only way to genuinely prove a cause and effect relationship, rather than just noticing 2 things happen to occur at the same time.
Now, to give you the rigid sequence required for those standard test questions, the steps are 1. Recognize the problem, 2. Observe, 3. Hypothesize, 4. Deduce, 5. Experiment, and 6. Summarize and report. You will definitely need to know this order front to back.
Okay, let's hit pause on the rote memorization for a quick second. You can't just throw things in a beaker and call it science. The real complexity is setting up a valid test. Biological systems are incredibly messy. Individual variation among living organisms creates a ton of background noise. So how do we prove our if-then statement without our own biases or environmental noise completely ruining the results?
That brings us perfectly to an exam staple, the control group versus the experimental group. In science, experiments must be controlled. The two groups are treated exactly alike, except for the one specific variable being tested. For example, if you're testing if carbon dioxide is needed for photosynthesis, you have a control group, a plant with freely available CO2, and an experimental group, a plant totally deprived of CO2. If photosynthesis only happens in the control group, well, your hypothesis is supported.
And that is the perfect textbook example. But here's what actually happens behind closed lab doors to ensure validity. We don't just have one simple control. We often use positive controls where we expect a known result just to make sure our equipment is even working, and negative controls to identify false positives. We use randomization to assign subjects to groups to eliminate systemic bias. We use replication, repeating the exact same test multiple times because, well, biology is unpredictable. And critically, we use blinding, meaning the researcher taking the measurements has no idea which sample belongs to which group. This literally prevents us from unconsciously skewing our own results to fit our favorite hypothesis.
Section 4. Gathering and analyzing data. Making sense of the numbers. Once that meticulous design is fully in place, this is where the experiment yields its results. To see if the hypothesis actually holds up, a biologist gathers quantitative data. Going back to our photosynthesis example, you'd measure the exact amount of glucose produced or oxygen released in both the control and the experimental groups. You're looking for a uniform or constant fact of nature here.
But again, raw numbers on their own, that's just noise. One plant growing taller might just be a random fluke. That is exactly why biomathematics and statistics are so integral to the scientific process. We don't just look at individual plant heights, we calculate averages, ratios, and probabilities. Statistical analysis is absolutely crucial to prove the findings have true significance, meaning the result is genuinely because of the variable we changed and not just some biological randomness.
Section 5. The role of skepticism. Doubt in science. That rigorous mathematical proof leads us perfectly into this concept.
For your definitions, make sure to note that skepticism is an attitude of doubt. It actually acts as a primary driving force behind the entire scientific method. It's an essential tool for objective inquiry, making sure that researchers really push themselves to rigorously investigate claims rather than just accepting them at face value. I gotta say, this is my favorite part of the whole process. Skepticism means scientists don't just automatically trust each other's work. When someone reports their results, the scientific community doesn't just stand up and applaud. They actively try to break the findings. They demand rigorous peer review. They literally try to replicate the experiment themselves to see if they get the exact same results. They force alternative interpretations of the data. It is a brutal, highly demanding process, and it ensures that only the absolute most robust, well-tested explanations actually survive.
Section 6. Scientific theories and laws. Establish knowledge. Let's see what happens to those explanations that do survive.
Here is your ultimate exam cheat sheet for a very, very common test question: the difference between a theory and a law. A scientific theory is a hypothesis that has stood the test of time. It's been tested repeatedly and never rejected, and it explains the why of natural phenomena. A scientific law, on the other hand, is a uniform, constant fact of nature. It describes what happens under specific conditions, and it is considered irrefutable.
To really ground these concepts, let's look at some famous textbook examples. Darwin's theory of evolution and the cell theory explain vast, complex mechanisms of why biology works the way it does. But Mendel's laws of inheritance or the Hardy-Weinberg law, those are established, definite entities that describe exactly what happens. And here's the massive takeaway. While theories are strongly supported, they can still be altered or refined if completely new evidence comes to light. Laws, however, are essentially set in stone for the conditions they describe.
Which leads us to a fascinating final thought for this explainer. We've seen the strict rules and definitions required to understand the biological method.
And we've seen the rigorous, messy, and deeply skeptical reality of how those rules are actually applied in the laboratory to carve out the truth.
So we'll leave you with this. If even the strongest scientific theory is fundamentally just a hypothesis that has managed to stand the test of time so far, what incredible new evidence might be discovered in a lab tomorrow that could completely rewrite the biology textbooks we rely on today? Keep questioning, keep learning, and thanks so much for joining us.